Welcome to Stanford Cancer Institute's Breakthrough in Cancer Seminar featuring Dr. Charles Sawyers from Memorial Sloan Kettering Cancer Center. We're delighted to see you all today, both in person and online. Our Breakthrough in Cancer Seminar series is the second Tuesday of each month at 4 p.m. with a reception to follow. And we have an impressive schedule for the rest of the year, and we hope that you'll join us for the next in the series as well. So September 12th, we will have Dr. Ursula Matalonis from Dana-Farber Cancer Institute, and her seminar topic is Drug Development for Ovarian Cancer, Challenges and Successes. So the format of today's seminar, we will have 40 to 45 minutes for Dr. Sawyers' talk, and then 10 to 15 minutes for question and answer. Charles Sawyers received his BA from Princeton University in 1981 and an MD from Johns Hopkins School of Medicine in 1985, followed by internal medicine residency at University of California, San Francisco. He became a Howard Hughes Medical Institute scholar in 2002 while at the University of California, Los Angeles, and then moved to Memorial Sloan Kettering in 2006, where he currently serves as the chair of the Human Oncology and Pathogenesis Program. Dr. Sawyers studies mechanisms of cancer drug resistance with an eye toward developing novel therapies. He co-discovered the anti-androgen drug enzalutamide that was approved by the FDA in 2012 for the treatment of advanced prostate cancer. He shared the 2009 Lasker-DeBakey Clinical Medical Research Award for the development of the ABL kinase inhibitor imatinib for patients with chronic myeloid leukemia, and the second-generation ABL inhibitor dasatinib to overcome imatinib resistance. He received the 2013 Breakthrough Prize in Life Sciences and the 2013 Taubman Prize for Excellence in Translational Medical Science, 2017 American Cancer Society Medal of Honor for Clinical Research, and the inaugural Stat Biomedical Innovation Award in 2019. Dr. Sawyers is a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. He's past president of the AACR and the American Society for Clinical Investigation, was appointed to the National Cancer Advisory Board by President Obama, and has served on the board of directors of Novartis since 2013. He serves as a steering committee chair of AACR Project Genie, an international consortium of cancer centers that share genomic and clinical data from patients treated at their respective clinical sites. And with that, I'll welcome up Dr. Sawyers.
Thank you, Allison, and it's great to see you thriving here in Palo Alto. It's great to see a lot of friends from various parts of my career. You said 40 to 45 minutes. I may exceed that a tad. I got a lot of cool stuff I want to talk to you about, and I do want to take a lot of questions. And I guess there's an hour reception afterwards, so who cares? Here are my disclosures. So I'm gonna try to tell three stories. The first is about plasticity, which is the end stage of prostate cancer, at least as how I conceive of it. It's a response to treatment. And then I'll begin, and I'll have two other shorter stories on prostate cancer initiation, which is something that I think is equally important to study. And I want to just frame prostate cancer for you. Okay, so this is the map of the oncoprint of primary prostate cancer, and the only thing I want you to take away is that roughly half of prostate cancers in U.S. or European men are caused by a translocation involving ERG or cousins, ETS family member cousins, and they are mutually exclusive with pretty much everything else except PTEN. And that's relevant for the last vignette I'm going to tell you. To set the stage for the plasticity concept, this is a plot from a large consortium that Arul Chennaiyan and I led some years ago to sequence metastatic prostate cancer at scale, sort of to go with the TCGA, which was all primary prostate cancer. And there's a ton of information in here, but I want you to see two or three things. First is that 50% of metastatic prostate cancers have one of these three, and often all three of these alterations: loss of p53, loss of PTEN, an amplification or mutation in the androgen receptor, which is, in this case, almost never seen in primary cancer, as you can see here, because it's sort of next to the 0. And that's because this is clearly a response to treatment with hormone therapy. The other one I want to point out is loss of the retinoblastoma gene, which is seen in about 30% of late stage and pretty unusual in primary. All right, so that's the genomics that you need to have in the back of your head as we go through this. Here's the clinical scenario in which prostate cancer is managed. It's detected early, often now, particularly in US, Europe, and is treated with surgery and radiation. And the cure rate is probably 60, 70%. There's a relapse rate of micrometastatic disease that wasn't initially appreciated at diagnosis. Or sometimes men present with metastatic disease. That's almost always treated with hormone therapy, and it almost always responds. What we used to call primary castration therapy, and this is called castration therapy because when a relapse develops, it's known as castration-resistant disease. So there's been enormous progress in the development of next-gen anti-androgens. Allison mentioned that the drug enzalutamide came out of work from our lab, and it's now actually used often frontline together with abiraterone, so these therapies are even earlier now. And so what we now have is a situation where men are living much, much longer with these more potent ways of inhibiting the androgen receptor, but we still have acquired resistance, and there's basically two categories. One is, despite all of this, it's still possible for the androgen receptor to further amplify or to create other mutations or to create splice variants that allow it to escape. And that's led to large efforts by many groups, including companies, to develop next-gen anti-androgens that would focus on AR degradation. So far, no home runs yet. But what I want to transition to now is this mechanism number 2, which we call lineage plasticity. We came upon this some years ago because we were seeing this as a new mechanism of escape. New meaning maybe it existed a little bit in the past and was not so well appreciated, but now seems to be more prevalent. Even though men are living longer, when they do relapse, they're relapsing without restoration of androgen receptor signaling or without a rise in prostate-specific antigen. And you actually can see this now, sort of once you know about it and start to look for it, you can even see this rarely at diagnosis. And this is from a colleague, Yu Chen at MSK, and Anu Gopalan, who is our GU pathologist who read the biopsies. So this is man unfortunately presented with primary localized disease and got the typical multi-quadrant biopsy. And we routinely sequence pretty much every patient who comes to Sloan Kettering now with a gene panel. And this patient had a pretty high-grade adenocarcinoma with P53 and P10 deletion and the ERG rearrangement. But at the same time, but in the other side of the prostate, he had this histology, which is small cell neuroendocrine-like disease with exactly the same genomic profile, but now with RB. So this is just, again, a rare case, but I think it's sometimes a patient like this is instructive because you can pretty much see what's going on right in front of your eyes. So we want to understand how this can happen. Clearly, they're clonally related. So we published a paper last year, which I'm going to review the highlights of in, say 10 minutes max, and then I'm going to go to new data that's on this topic as well as the other two topics. So this is a cartoon to lay out the flow of a series of experiments that were in this paper. And we sequenced patient samples from late-stage disease, but I think the real clue or tool to help us understand how this happens is to take advantage of a mouse model that we had collaborated with the Goodrich lab in Roswell Park in characterizing a couple of years earlier, and that is deletion of the 3 major tumor suppressors that I pointed out to you on that earlier slide. And what happens in these mice is that they develop an adenocarcinoma, and they become castration resistant. And as you will see, when we use single-cell analysis to characterize this, we get all kinds of different types of histologic forms of progression. So I'll show you, so basically the layout of the experiment was to take wild-type mice, what we call a DKO, or tumor suppressor gene knockout, or the TKO, across a time course for each strain that was picked based on the kinetics of disease that we knew developed from characterizing it morphologically. And in the wild-type mice, what you see, as we had previously known from a single-cell analysis of just wild-type prostate, is 3 epithelial populations, 1 large basal cluster and 2 distinct luminal clusters, which we have called L1 and L2. And I won't get into the specifics of any of that for the sake of time, but this is a stem-like population that's much rarer than the L1 population, which is the more secretory luminal cells that, at least in the human, make PSA. And what you can see in the 2 models, if you go through time, and this is the faster model, you can see that very early after deletion of the tumor suppressors, you lose these normal populations, and your epithelial population now shows up here, and it's a classic adenocarcinoma. And then you start to see these subpopulations appear with increasing, sort of number, and then with the TKO, with an NEPC component that comes out as early as 12 weeks. So we interpret this, although we don't have a formal lineage trace to prove it, is that this population here is the source for all of this plasticity. We have lots of computational data to point to that conclusion. And as you'll see, we have organoid-based data to get a little more granular on that. But many questions arise. One of the most urgent ones was, what is the transcriptomic profile of this population, which I was sort of imagining as this cauldron of potential plasticity. And then sort of simplifying a huge amount of analysis in this one plot. Here's signatures that are highly enriched in this adeno population, as designated by red. And if you just read across them, you will see that it's a whole lot of signatures involved in inflammation or inflammatory signaling writ large, including JAK-STAT signaling, interferon response, etc. And curiously, if you look, I'm only showing you NEPC here, but if you look at those other subpopulations, it's no longer active once cells have sort of committed to those new lineages. You can look at this in more detail. This is focusing on a series of transcription factors that we're assigning as highly expressed in the adeno state or the neuroendocrine state, and then a series which we're calling transition transcription factors, which include actually STAT proteins that would presumably potentially be linked to the JAK-STAT activation. So this is all really fascinating, sort of description of what's unfolding over time in these mice. But we obviously wanted to get to, is it doing anything? And also, what is the source of the inflammation? I didn't tell you, but there's an enormous inflammatory infiltrate coming in early in these tumors, which would be an obvious explanation. So it turns out in other work in the lab, we had perfected the ability to grow these mouse prostate tissues as organoids in a collaboration many years earlier with Hans Clevers. So we took that approach and tried to model this whole thing in organoid culture. So this is, so what we do is we take the normal prostate out of a mouse that has floxed for the relevant tumor suppressors. And at least in this rendition, that's how I'm going to show you. But as I'll get to in a minute, we can do this any way you want, CRISPR, et cetera, ex vivo. But I'm not taking this prostate out of a mouse that has prostate cancer. The mouse has no Cree driver. We're developing a normal prostate organoid and then infecting with a Cree virus and watching what happens. And what happens is the lumen fills in, the polarity between basal luminal cells is lost. In a lot of detail in the paper, we show by single cell analysis that there's sort of a mixing of basal luminal identity. A little bit of mesenchymal type features with cells that start to migrate out of the organoids. And this happens, it takes time for this to happen. We start the Cree deletion here. I won't show you, but the Cree deletion is extremely efficient, and there's the data documenting that is in the paper. And it takes about 4 weeks, and it's almost like clockwork. Suddenly, they start to fill in. So there's some process going on that obviously we're quite interested in. It's in the paper, but we did RNA-seq, and the inflammatory signaling pathways that I showed you from the in vivo are all on in this very reductionist system where there are no inflammatory cells. So this is a cell-autonomous activation of those pathways. The next question is, can we reverse this? So we took advantage of the obvious inhibitors that exist. And without explaining why we also included an FGF inhibitor, other than it showed up on some of our transcriptomic analysis, what I've boxed in here in red is what I want you to hopefully see very clearly, and that is these organoids which whose cystic morphology is completely lost because of this loss of polarity and filling in, is completely restored within 2 weeks of treating with these drugs. And this is all quantified here. What we also see is that the androgen receptor, which is expressed in these organoids and is expressed here but not as high, is much higher expressed here, and the organoids are resensitized to anti-androgen therapy. We also see that in a subset of human patient-derived organoids, that Yu Chen had developed. Here's an example of one which is expressing a little bit of androgen receptor in some cells but not all. And then when we treat with the with the FGFR plus JAK inhibitor, we greatly increase the level of androgen receptor expression and get morphologic changes that make them more luminal. So there's a lot more data in the paper showing the breadth of this response, which is restricted to a subset of castration-resistant prostate cancers, and it's a subset that can be ascertained by RNA-seq analysis for seeing activation of JAK-STAT signaling. A cartoon that sort of gives the punchline of where we left off at when we wrote the paper is that when we lose these tumor suppressors, we create this mixed lineage state. And what I'm now going to show you is that although we don't see this in organoid culture, when we put them into animals, we can get the full range of plasticities that we saw in the mouse. But if we try to intervene with these inhibitors, if we intervene at this stage, we can get them to go backwards. But if we intervene at this stage, it's too late, at least for that strategy. So with that, I now want to move to new ways of interrogating the same question, but having more sort of experimental flexibility to play with many more variables and move more quickly. And this is the work of Rodrigo Romero. And so now, instead of relying on mice with the right flox alleles that you've bred to the right configuration, we've taken advantage of sort of what may be a bit of a lucky break in prostate organoids, and we've not tried this more broadly. But using the RNP method of transfection or electroporation to introduce Cas9 protein with the relevant guides, we get incredibly efficient deletion of genes of interest. And we can do it in multiple combinations, not for the sake of time not showing you, but we can make very large deletions. It's extremely efficient. And it's really changed the sort of the workflow in the lab. We can also infect with lentiviruses expressing genes of interest. We can tag them, et cetera, and we can put them back orthotopically or in other places in the mouse. So I'm just going to now give you a sense of what we're learning from this by playing with a whole suite of different genes of interest. So I'm just going to, we've done an allelic series of the 3 main tumor suppressors that I showed you now in singlets and all the possible doublets and in triplet. And basically the rules are that if you have a combination and you want to see neuroendocrine prostate cancer, the combination of P10 and P53 does not get you there, but the combination of RB and P53 does. Furthermore, as I sort of hinted at a second ago, you do not get it if you grow them purely in organoid culture, in matrigel with standard, serum-free ENR conditions, EGF, noggin, R-spondin. But if you implant them into mice, orthotopically, or as we've now subsequently learned, even subcutaneously, in the case of the RB deletion complex with P53, you get an adenocarcinoma that then transitions to a neuroendocrine cancer. And the histology of this, I'll show you more of, is absolutely spot on, and all the molecular markers are spot on for what we see in the mouse, the GEM model, as well as in patient samples. Conversely, if you do this with RB intact, you get adenocarcinoma, and that's all you get. And these are beautiful adenocarcinoma models. Here's a little bit more of how this works. We also, I should say, we're doing this in a purely, in a syngeneic background, all black 6. There's a whole immune component that we can study, and I won't really have time to tell you much about where we're going with that unless you want to ask. But I hope you can see, Paul's a pathologist, I know, and maybe there's some others in the audience. And I've spent a lot of time looking at mouse models of prostate cancer. This is as good as it gets in 30 years of doing this. So we're really pleased with what it looks like. And then we can see these transitions take place. We get metastasis in this model. We can track them with the GFP, et cetera. To sort of, confirm what I told you a minute ago in terms of markers, perhaps this is the best way to see in the RB loss model the acquisition of ASCO1 expression, DLL3, the cell surface marker, which goes hand in hand with ASCO1 and is of great therapeutic interest, synaptifies in NCAM, whereas you don't see that in the P10 model. Eventually you see complete loss of androgen receptor expression in the RB model. So now that we have this, we're doing all kinds of things. I want to just give you a taste of some of the spatial analysis that we're doing. So this is a platform called Lunaphore Comet, which is basically multiplex immunofluorescence. And this is one section of one of these orthographs, and you can, and we've, this is the H&E, and you'll just have to take my word, despite the low power, that this is NEPC here and NEPC here, and in the middle is adenocarcinoma. And here's some of the immunohistochemical evidence for that. This is keratin 8 staining, which is a luminal cytokeratin marker, and here's ASCO1 staining here and here. Just in terms, there's many things to obviously analyze here, but one quick takeaway, if you really focus down here, is that CD8 T cells are completely excluded from the neuroendocrine regions, but retained in the adenocarcinoma regions. This is not necessarily a new finding. I think we know this from some of the human clinical data, and those of you who are immuno-oncologists know that prostate cancer has been a huge disappointment with checkpoint therapy. Whether this is going to be informative for that, time will tell. But what I think is perhaps surprising is that there's actually a significant myeloid infiltrate that's present in the neuroendocrine population. And these cells type as CD11C positive myeloid cells, which, depending, and I'm not a myeloid biologist, but I know this is a marker often for dendritic cells. Perhaps if we see these kinds of things in patients, which would have been maybe perhaps difficult to discern without having this kind of spatial technology, it could lead to some new ideas for immunotherapy treatment of prostate cancer. What I think is perhaps more sort of instructive in terms of this analysis is we're able to ask, or at least begin to ask the question, where does the neuroendocrine transition emerge from? Where is the sort of birth of this subpopulation happening? And what I'm trying to show you here in these images is that it looks like it's coming from the luminal CD8 positive cell and not the basal CD5 positive cell. So what I hope you can see is that the ASCL1 positive cells are often double positive with keratin 8, but not with keratin 5, where they're sort of adjacent. This has been quantified across a large number of sections here, in which there was large, as you saw, there were populations that were just pure NEPC. That's this large sort of 80% slice of the pie. The ones that are keratin positive for either 5 or 8 and ASCL1 are mostly keratin 8 positive, and there's more quantification there. So another thing we've been starting to do is use transcriptomic, spatial transcriptomics using the Visium platform to examine these sections. And this is another section showing in which the NEPC portions are separated from the adeno portion here. And I hope you can appreciate that these 3 NEPC regions all uniformly express ASCL1, which we know is true from the lunaphore I just showed you, as well as more conventional immunofluorescence. But what I've boxed in, so this is a set of transcription factors that were chosen based on some single-cell RNA-seq data as being differentially expressed between neuroendocrine and adeno. And what I've boxed these 2 others because it points out that they're not uniformly expressed in every region. So I hope you can see that the SOX13 is primarily here, but not as much here, whereas the OLIG3 is here and a little bit here, but not here. So although we think of, at least I was thinking of this neuroendocrine subpopulation as being sort of a clonal, sort of bottleneck-driven outgrowth from the selective pressure of treatment, once it gets there, I think it now opens back up into greater heterogeneity. But it then, begged the question, well, what if we, can we stop this? And as I tried to, tell you with the sort of the not plasticity, that's not the right word, the manipulability of the system, we have the ability to move quickly on functional perturbations. So in a relatively short period of time, Rodrigo was able to just say, let's play the movie again, let's delete the tumor suppressors, and let's now include or not an ASCL1 guide and see what happens. And we're going to transplant subcutaneously and orthotopically in this experiment. And the result is absolutely crystal clear. Even though the cells don't express ASCL1 when they're in organoid culture, if you delete it then, there's no way that they can form a neuroendocrine outgrowth. And in fact, these are sub-Q tumors now, and this, again, is, I love histology, and this is just a beautiful adenocarcinoma. A lot of sub-Q tumors, some of you may know, they start to get sarcomatoid and all kinds of funkiness. This is remarkable. And what's also shown here is that's retained in castration. But the fact that this is such a beautiful adenocarcinoma, and I'm not showing you for the sake of time how much androgen receptor is now very highly expressed here, but not here. They're very castration sensitive. And that's shown here. So these sort of dark blue lines here, this is the summary of the data. These are the individual mice. They barely form anything. And these are the castration subgroup in which ASCL1 has been deleted. There's a few other points here. The ASCL1 intact arm, the red and the yellow or orange, the castration in that setting accelerates the growth of the tumors. And that we're almost 100% sure is due to accelerating the rate at which the transition to neuroendocrine happens. And we've quantified the proliferation rates of the neuroendocrine subcomponent versus the adeno, and the neuroendocrine grows faster. And that's very consistent with clinical anecdotes that are starting to emerge from patients who start to undergo this transition and have very rapid disease progression. And finally, on this part, I want to make another point, and that is ASCL1 deletion completely blocks the metastatic phenotype that we see when we do orthotopic implantation. You know, it's still a small total number of mice, 12, but it's absolutely flat out 0. So you can draw all kinds of, next step conclusions, but I think ASCL1 is sort of the gatekeeper transcription factor for this transition. We're very much interested in what step when it goes in vivo leads to it turning on. Is it receiving a microenvironment signal? Is it losing a signal that was present in organoid culture? I think we're going to figure it out hopefully soon. And can we intervene? So these are the takeaways so far. I won't repeat them all. I'll just let you look at them for a second, and then I'll move on to the other 2 vignettes. So I've spent all this time talking about metastasis and the response to castration therapy and the plasticity that can emerge. What I'm now going to talk about is the very first steps. So I showed you in that first introductory slide that there's a set of early events. They happen in the cases in which it's been carefully analyzed, they can be detected at the stage of what's called PIN, or prostatic intraepithelial neoplasia. For those of you who are familiar with pancreatic cancer, this is sort of the Panin equivalent. And the definition of moving to invasion is breaking through the basement membrane. Now, I need to just acknowledge that there is some still debate as to whether PIN is truly a precursor lesion or just something else that is a dead end. And I'm not going to argue that either way. I just wanted to show you this conceptually to make the point that invasion requires progression beyond the basement membrane. So how was one start to look at this? Well, we again turn to genetically engineered mouse models, and this actually is a model that my lab made more than 20 years ago when I was still at UCLA, which is a very simple model. It's the old style, making a transgenic by pronucleus injection. In this case, the transgene is MYC, and we got 2 stable strains that we fully characterize. One that expresses high levels of MYC due to the number of integrations, and one we call low MYC. They both get PIN, and then they get invasive prostate cancer. This one's faster, so this is what we used for this study. And I'm showing you the kinetics of 6 weeks, which is pretty much the earliest that you can really detect transgene expression. You see PIN. So it's very fast display of the phenotype. 6 months goes by, and then it's still PIN, early signs of invasion, and then by 8 months, pretty much every mouse has invasive adenocarcinoma. So we did a time course, single-cell RNA-seq profile to try to get clues as to what was happening during this transition. And here's the UMAP of everything, including the littermate control mice. And you can see that this is the luminal population, L1 and L2, and all the transgene expressing cells are here. There's a large myeloid infiltrate here that you can't tell from the way this is labeled, but it's mostly only seen in the transgene positive mice and not the wild type mice. Those are the 2 biggest things that jump out from the overall view. The next question we asked was, okay, you undergo this incredibly dramatic histologic transformation from early stage PIN, later stage PIN to invasive cancer. what does the UMAP look like just for the transgene positive epithelial cells? And that's these clusters here. And you'll just have to sort of look at the colors and take my word for it. But if you map across all the 4 time points that we sampled, there's no enrichment across any of these clusters. And we've got all the quantification for that. So basically, at a UMAP level, the transcriptome of the tumor cells is identical, whether it's encapsulated or widely invasive, which is the opposite of what we saw on the left, if you remember, from the tumor suppressor deletion model. So it raises the idea that something else is happening. Is there a signal from the tumor microenvironment that's causing this switch to invasion? So I mentioned the myeloid cells. To get more specific, we did both flow cytometry and immunohistochemistry. And I'll just point you to this AIF1 stain, which I hope you can see here. You can see a little bit of brown in 6 weeks. You see pretty convincing within these glands at 3 months when there's no invasion at all. And you have to look over to the right to see that there's nothing in the, I'm sorry, you have to look up here to see that you don't see that in the wild type mice. And then of course, as the tumor progresses to an invasion, you get a really just heavy myeloid infiltrate, which obviously could be a wounding response. So chicken or egg, what's happening first? The timing of this says the myeloid cells are getting there before the invasion. But of course, you have to do the real experiment or the right experiment, which is to see if you can deplete them and see if you perturb the invasive phenotype. So we did this using, and there was more characterization that told us that these were primarily macrophages. They expressed high levels of CSF1 receptor. So we used the experience of vast literature and other investigators to use this antibody to deplete the macrophages. And we specifically picked this time period. You can do this for a certain amount of time experimentally before you sort of run out of rope because of immune responses to the antibody and so forth. But we picked that 6-month time point because we were very confident in the kinetics of the model, that we'd see very little invasion then, and we wanted to see what happened. So the rest of this slide is just showing that we can deplete from the prostate successfully a large fraction of myeloids, of macrophages and myeloid cells. The really exciting day in the lab was when the first cohort was sacrificed, and just by gross morphology, these are prostates that have been removed en bloc at the time of sacrifice. You can see in the isotype control mice these nodules all over the prostate. That's invasive cancer, and you just don't see them in the CSF1 receptor antibody treated group. The prostates don't weigh as much, but is that because there's is that how much the myeloid cells weighed? It's more than that, obviously. If we look histologically, you can see in the isotype controls, there's a widely invasive cancer here. A really nice stain in scoring mouse prostate cancer is this smooth muscle actin, which stains the rim of fibroblasts called myofibroblasts around each gland. They're just obliterated when you have invasive cancer, and they're still retained for the most part in the mice that received the CSF1 receptor depletion. And Michael Hafner at the Hutch scored a whole set of these slides in a blinded way. And he scored it in 2 ways. One, is there any invasion present at all in multiple sections of the prostate? And that's this analysis. And we don't completely prevent it, but it's definitely reduced. But if you look at sort of the surface area of invasion across the slide, it's a very dramatic effect. So why did this work? There's an enormous literature, I think, as many of you know, about myeloid-derived suppressor cells and macrophages impairing what would be a normal immune surveillance function of CD8 T cells. So in order to see if that was the mechanism, we decided to deplete the CD8 T cells and also deplete the macrophages and see if we rescue the phenotype that I just described. So we set it up exactly the same way, same algorithm here of treating during that 6 months to 8 month time period. And you can see by prostate weight that we get a big effect with we repeat what we saw before, and we do not rescue by CD8 depletion. I guess shown up up here is the fact that we do deplete CD8 T cells from these mice. So this is not absolute proof that it's independent of T cell function, but it's suggestive that something else is going on. So we went back and looked more carefully at the expression profiles of the macrophages. And that here's now focusing in on a UMAP of just the myeloid cells and naming them as tumor-associated macrophages or tumor-associated neutrophils based on their presence only in the transgenic model and their expression profile. And whether you like it or not, you're almost obliged to test for the polarization signatures of M1 versus M2. And the answer is it's like neither or both, depending on your sort of in between. If you do differentially expressed genes, one of the genes at the very top is interleukin-1 beta, highly expressed in the myeloid cells in the transgenic animals, beginning at the earliest stages of infiltration. It's not, IL-1 beta is part of the M1 signature, usually with IL-6 and other inflammatory cytokines, but IL-6 is not expressed. So I don't think this is just classic M1. There's something different here, but this caught our attention for many reasons. I mean, the data just pointed right there, and also, as many of you know, IL-1 beta is getting more and more interesting as a potential target for drugs. There is a clinically approved antibody that neutralizes IL-1 beta, and the patient population are individuals who have familial inherited fever syndromes due to defects in inflammasome components. It's a very successful drug. It happens to be a drug that Novartis makes, which is, I'm on the board of Novartis, so there's my conflict. But it's another reason I knew about it, and I thought we should chase this. There's also a set of data that came out of a clinical trial for cardiovascular disease or an atherosclerosis risk in patients who had already had a cardiac event, in which IL-1 beta neutralization with that antibody reduced the frequency of a second cardiac event, which is exciting in that it's pointing to that mechanism of inflammation in atherosclerosis. But there was a completely unexpected finding in those patients, which is the incidence of lung cancer, new lung cancers over the 4-year period of treatment was dramatically reduced in the group that got the IL-1 beta. So is it possibly preventing lung cancer in high-risk individuals? So for all those reasons, we wanted to go deep on this. So we went straight to the same strategy, this time using IL-1 beta neutralization here. And the answer is remarkably clean. We see very little invasion just by neutralizing IL-1 beta, phenocopying what we saw by depleting the macrophages. So why does this work? Well, I'm going to show you two things that we think explain this. One is, I hope you're convinced we have pretty direct evidence that IL-1 beta made by macrophages is talking directly to the tumor cells and inducing proliferation. And the other, which I didn't expect to find or wouldn't have imagined, is that IL-1 beta is regulating the expression of matrix metalloproteinases, primarily by macrophages, but also tumor cells. So start with the first one. First question is IL-1 receptor expressed on the tumor cells? And the answer is yes. For the sake of time, I'll just ask you to believe me. Here's really bright staining for IL-1 beta, IL-1 receptor in the proximal region of tumor-bearing cell of transgenic mice, as well as in the normal mice. But if you look at the distal region, which is also regions where tumors can develop, you see high expression here, but not in wild-type mice. You can also, at least in my mind, it's still not exactly clear what the best sort of downstream readout of IL-1 receptor activation by IL-1 beta is in a tumor cell. But we looked at many things, including P38, based on the literature. And so we see pretty impressive phospho-P38 here. And if we treat with the neutralizing antibody, we see that go down substantially, as shown here compared to here in the isotype control. So that's correlative evidence for direct, IL-1 beta to tumor cell stimulation. We can use organoid culture to get, a little more specific, and we did that in two ways. First, we made bone marrow-derived macrophages and supernatants, which include IL-1 beta, and showed that they can stimulate the growth of organoids derived from the transgenic mice. But then if we use recombinant cytokines, and we used a set here, IL-1 beta clearly works at all these time points. So that's mechanism one. The other mechanism came serendipitously from a collaboration with Sanjita Bhatia's lab at MIT. Sanjita had reached out to us some years ago and asked for the HiMic mice to test a new probe that she had developed to measure protease activation in tumor cells. And this is the way this probe works. It's got a fluorophore that's released if this linker is cleaved, and you can make linkers that are specific for different classes of proteases, and this is an MMP-specific linker. And she showed in this paper that the HiMic tumors, there's a lot of protease activity, whereas there isn't in the, and then this is the blocking agent for the probe. So having now seen these effects with IL-1 beta and with CSF1 receptor antibody, we repeated these experiments with her. And what I hope you can see is that the activity of the probe is there in the isotype mice, and it's greatly diminished if you deplete the macrophages or you neutralize IL-1 beta. And of course, we can use the single cell transcriptomic data to hone in on which, by the way, this is an MMP based on the linker. It turns out it's most likely MMP19 based on the very high expression of MMP19 in the tumor-associated macrophages in this population here. And then a week after IL-1 beta neutralization, we see a decline in the expression of the MMP19. So to wrap up this vignette, we think the invasion phenotype in this model is absolutely critically dependent on the myeloid infiltrate, which is bringing in IL-1 beta to further stimulate proliferation and to get invasion, the protease effect through MMPs. We have to, of course, prove that the MMPs are doing that, and that'll obviously be next. So this is the summary here, and I'll just let you look at it for a second. And I've got 12 minutes left to do the final little vignette. So now we're going back to the beginning, which was the ERG translocation, present in half of men in the US and Europe who have a diagnosis of prostate cancer. So the reason, so the thinking about this translocation is that ERG is not expressed in prostate epithelial cells at all, zero. As a consequence of this translocation to this gene called TMPRSS2, it gets turned on because TMPRSS2 is very, very highly expressed in luminal cells. So it seems very likely that that event takes place in a luminal cell, maybe a luminal progenitor cell, that then is the cell of origin and gives rise to prostate cancer. So, but we know very little about why it's doing that or how it's doing that. We and others have generated lots of differentially expressed gene lists from ERG positive, ERG negative models. there's differences. It's all very interesting, but it hasn't really told us the answer. So having now you've got the theme of having a gem model, throw single cell seq at it, and see if you can learn something. Here's vignette number 3 on that. So this is the work of Wei-Ran Feng together with Christina Leslie and Eric Ledwig. And we made this mouse some time ago. It's a mouse that expresses ERG downstream of a Rosa lock-stop blocks cassette. If you cross with P10, Flox P10, and then cross with probasin Cre, which is expressed in basal cells and luminal, all androgen receptor expressing cells basically, you get a beautiful model of prostate adenocarcinoma, which we characterized at 6 months when nearly all the mice have invasive prostate cancer. But Wei-Ran looked much more carefully earlier, and he could see foci of prostate cancer as early as 3 months very convincingly, and you don't see it with P10 alone, where you just see these sort of introductal lesions. So single cell sequencing analysis of that time point, really focusing deeply there, results in this UMAP. And there's a ton of data here, of course, as well, but for the sake of time, I'm going to draw your attention to the population of cells that's proliferating. And that's this cluster 10 and a little bit cluster 1. None of the rest are really proliferating, and here's the proliferation signature. So, our assumption is if you're going to go where the money is, go where the proliferation starts early after activation of the oncogenic event. And what surprised us is that when you now do lineage assignments using transcriptomics, these are basal cells, which went against everything we thought. They should be luminal because of temporous expression, et cetera. But there it is. And you can see the ERG expression there. Now, this is a probasin promoter, so it's not restricted to luminal cells. But but it is very highly expressed in luminal cells, so the gene is clearly deleted in luminal cells. But the cells that are responding are basal cells, and I'm calling them basal IM because they actually express a little bit of luminalness, but on a UMAP, they don't move enough to be called luminal. So here's the histology of the tumor when it develops is purely luminal. So here's a cartoon to lay out what we think is going on. The transgene is activated here, but quickly the cells acquire a luminal fate. And that for a period of time, the proliferation is in a cell that's kind of confused. It's both. Before it fully commits to luminal. I just put this up here because I wanted to argue that people are still arguing about cell of origin, and I don't want to get into it, but we now had to at least do a few experiments. So first, let me just convince you that one of the things ERG does, besides activate the proliferation in this basal-like cell, is to make it very quickly go luminal. We did EDU tracing to show that, which I'm not going to show you here for sake of time. But what you can do is you can isolate fresh prostate from an ERG P10 mouse with no Cre driver. You can separate basal and luminal cells using cell surface markers and plate them separately, and then activate or add Cre virus and turn on the process. And what I hope you can see is that you really increase the amount of luminal cells that appear with just 2 passages compared to wild-type mice. P10 alone does it to a certain degree. If you start with luminal cells, they quickly crash. This may be true physiology of terminally differentiated secretory luminal cells, or it may be our shortcomings in the way we culture these cells. Another way to ask this question is to knock in fluorescent markers downstream of basal and luminal lineage markers, keratin 5 and keratin 8, and then separate based on color live cells and plate them. And here you can really see the effect of ERG. This is ERG, and this is P10 only with ERG deleted. So there's no question that ERG drives this fate change. What about, forming a tumor? So we can take that same strategy of isolating the basal cells and the luminal cells from a fresh prostate, Cre deletion, and in the same day go back into mice of purified luminal Cre deleted versus purified basal Cre deleted. A bake-off, who's going to form tumors and who's going to form fastest, and what are they going to look like? And the answer is the basal cells form the tumors fastest, and the histology of the basal tumors is purely luminal. And they are clearly invasive, whereas the ones that form from purified luminal cells are not really invasive in the classic way. Then one last experiment on sort of to address this, because that's a reconstitution transplantation model, and purists can argue that, okay, that's a stress on the cells that maybe is not so physiologic. We used adeno-Cre virus with either a K5 or K8 promoter to restrict its expression. We went to enormous pains to validate that these truly are basal and luminal specific. And when you do that experiment, you get tumors from the basal K8 express Cre virus, and not from the K8. And when you do get an outgrowth, they're sort of intraductal. They're not invasive. And the ones that derive from the K5, the basal cells, are purely luminal. So I think we've sort of nailed that is the cell of origin in the mouse. But how do we turn this back to the question that I've started with is, what about temporous expression? So it turns out that temporous and NKX 3.1, which is another luminal marker for which there's a lot more Cre driver data in the prostate field, it turns out by looking more carefully, we found that they are both expressed in basal cells. I'm not showing the temporous data here, but here's using a YFP reporter in a normal prostate, roughly 25-30% of cells that are NKX positive are basal. And there's data to show that by spatially. Here's an experiment to ask if we don't have a let me back. We love a Cre driver that is specific for those basal cells shown here. We don't have one. No one has one at this point. One may not exist. But we can, and as you can see here, NKX1 is expressed in a huge fraction of luminal cells. But can we take advantage of the fact that these cells express K8, and I just showed you we can't get tumors from K8. Can we get tumors from NKX, and do they come from here? Well, the answer is we do get tumors from NKX Cre, and this is a tamoxifen-inducible allele, so we can really control the kinetics, and you can see the time at which the tumors develop. You can see the kinetics at which ERG-positive cells emerge. You see a certain fraction present 1 week after adding tamoxifen, and they expand quite significantly over the first month. So what's shown here is now flow cytometry analysis of those ERG-positive cells and what is their identity between 1 week and 1 month. And what I'm showing you here is that it's this basal, we interpret this as this basal population is expanding and becoming double positive. It's this intermediate proliferative population that I showed you on the first single cell map of the model. So finally, this is all mouse, do we see any evidence of this basal subpopulation that expresses temporous in humans? And the answer is yes, and we actually can see in human ERG-positive patients evidence of ERG expression in some of the basal cells. So I'm out of time, so I'll finish with this last slide, summing up vignette 3, which is we believe now that sort of contrary to what I would have thought, that the cell of origin is actually this subset of basal cells that look like basal cells. That's why they're drawn this way in this cartoon. And they undergo the translocation, and they start to proliferate, acquire luminal characteristics, and then fully go all the way to a luminal cell. And we think if we're going to figure out what's ERG really doing and how it's causing cancer, we need to isolate these cells and really study, where ERG is binding, what's the transcriptome here versus the whole tumor mass. And hopefully we'll figure that out. So here are the people. I mentioned them along the way. Here's the 3 stories, and thank you very much. And sorry I went over time. Yes?
Very exciting work. 2 of the commonly clinically used markers for prostate cancer, PSA and PSMA, you didn't mention at all. Are you looking at their expression with the transition and plasticity?
Yeah. So PSA is not expressed in mouse, so we can't do that in mouse, but PSMA is. Both of those are, I consider, luminal adenocarcinoma sort of lineage markers, and they gradually fade as this transition to a non-epithelial lineage develops. So for PSMA, it's of great interest because of the therapy that's now, again, a Novartis product. I didn't plan this, but the radioligand therapy. It's now approved for men who have late-stage prostate cancer. Its safety profile is very favorable, so it's being moved up earlier. Men relapse with late-stage, and I think they do so because the expression of PSMA is quite heterogeneous when you're late-stage and starting to undergo this plasticity. If used very early, I have great hopes for that therapy.
So the neuroendocrine component doesn't express PSMA at all?
There are examples where you can see it, so it's not a one-to-one, but in general, that's true.
So you explained the mammography just coming in as being a key step. So what is the signal that attracts them to come in, right? Because you have something happen in the epithelial cells, and all of a sudden they show up.
Yeah, there's a short list of cytokines and chemokines that have previously been shown to recruit myeloid cells, and they are expressed. We haven't shown that directly yet, but we can do it.
Fascinating. What's the relationship between RB1 loss and ASCL1? Do you have any sense of what's actually driving that interaction? Is there an interaction or the co-occurrence?
I think RB1 loss is required to precipitate upregulation of ASCL1, but it's not sufficient until you, at least so far, you have to go in vivo in our hands. We're working on other ways to try to get that to happen. What property of RB is doing that is your question. And all I can say is I think it has nothing to do with cell cycle regulation. So is there an E2F that's released that's somehow involved? I was hoping to talk to Julian, but he's away today. But we were emailing about this in the past because he knows a lot about this. You may know this. In the IPS, reprogramming world, the efficiency of, depending on what your favorite TF cocktail is to make iPS cells, is greatly accelerated if you lose RB. So there's a property of RB that is involved in chromatin state in some way that I would say creates a poised state that allows whatever this ASCL1 signal is to act. And if RB is intact, it can't receive it. Those are what I know. I just don't know the molecular basis of it. Maybe you do. Yeah.
Kind of a two-part question. So you mentioned in the normal prostate there's the basal and the L1 and L2. And I can't remember, you said one of them was more the progenitor, the L1.
L2 is the rarer and more progenitor.
So are basal cells not stem cells that regenerate with hematologic epithelium in the prostate? Like if you just are a basal cell, do they trace out and are they not stem cells?
If you do that experiment in an adult mouse, basal cells give rise to basal cells, luminal cells give rise to luminal cells. Those were not done with tracing alleles that were sophisticated enough to distinguish between L1 and L2, but it's L2. The reason I say that is because you can do another version of that experiment is to take adult mice and castrate them. The prostate regresses, and then you can give testosterone, and over 28 days it will fully reassemble. And the L2 cells persist after castration, and then nearly 50% of them within 24 hours of adding androgen go into cycle and give rise to this. So the basal cells probably in early development are a source. If you do the experiment differently and now go to pure reductionist system and take a single basal cell, drop it in organoid culture, it can give rise to a full organoid with luminal, and you can get the same from a luminal cell, but it's almost impossible. It's very rare to get it from an L1. It's much better from an L2.
So an L2 can generate basal cells and itself?
Yes, but in the context of a castration regeneration, where L2 does all the heavy lifting of regenerating the gland, basal cells aren't damaged by castration so much. So I don't know that. We didn't do those experiments with a lineage trace to know if there's any component there.
Thank you very much. Excellent talk. In your first vignette, you mentioned that sort of transitional state that the JAK2 inhibition and PFR inhibition could be reversed away from neuroendocrine differentiation. Do you think there'd be blood-based markers you could identify that in humans, or would that be something you have to do another biopsy to identify that transitional state?
Yeah, great question. Because I'd love to do a clinical trial, maybe you would too, with these kinase inhibitors. Actually, I wonder if Howard knows if this would work, but I'm interested in, you have to detect a cell state in the blood, I think. There's just no way to do this by imaging or biopsy today that I can think of. But I think it should be possible using either this, whole genome low-pass sequencing to look at nucleosome occupancy sort of patterns. I don't think it's sensitive enough based on some conversations I had with Gavin Ha at the Hutch, who's been doing some of this. But I wonder if methylation, DNA methylation, is going to give us at least a hint at that, and that's, as we all know, extremely sensitive. So there's a lot of people, not me, working on how to do that now.
Charles, we'll take the last question, an online question asking what is the interplay between ASCL1 and DLL3?
All I know is that ASCL1 is the transcription factor. DLL3 expression as a cell surface marker is absolutely one-to-one in our mouse models and in a human prostate TMA that Michael Hafner has worked on together with us. Is it a direct target gene? I don't know. It's of great interest, almost similar to the question about the PSMA radioligand. There's DLL3 antibody-based therapies that are more advanced in small cell lung cancer, where I think I forgot to tell you this. Everything we saw in this neuroendocrine transition is a exact phenocopy of the transcription factors in small cell lung cancer. But I'd say DLL3 is pretty darn interesting from what I'm hearing from clinical data.
All right, let's take a last second and thank Dr. Sawyers for an excellent presentation.